So without further ado, I would like to hand over to our first speaker. So Dr. Sepideh Khodaparast, who's going to talk about growth of myelin figures from parent multilamellar droplets. And she is in the School of Mechanical Engineering at the University of Leeds. So I'm going to drop my screen off and hand over to Sepideh.
So hi everyone, thanks for being here this afternoon for the LFD annual lectures. My talk is about growth of myelin figures from parent multilamellar vesicles, and unlike the rest of the talks of this afternoon, which are mostly probably going to be computational and theoretical modeling, this is a purely experimental work. And before I start, I would just like to briefly acknowledge the contribution of João Cabral and Will Sharrett from Imperial College London, and Robert Daglish, who is the instrument scientist at Larmor diffractometer at ICIS, who helped us with the SANS experiments. So there are a few keywords in the title of my talk, starting with the myelin figures, that I will start the introduction with. So what are the myelin figures? If you look at it's probably the origin of it is in the neurological and medicine and biology, where if you look at a neuron cell, it starts from a source and connects it, which it will be connected to the terminal through the axon terminals. So on the source side, it has the dendrites, it has the cell body and the nucleus, and it has a very long body of axon that connects the source term to the source body to the terminals, which is the destination where the signal will be transferred. And this axon can be as long as 1 meter, and its diameter is less than a micron or a maximum a few micron. And you can imagine that it's very fragile and it's very thin and long. So in order to be able to transfer the signals from the source to the destination as fast as possible without and as efficient as possible without any loss, is being covered by another structure, which is here depicted in blue, and is called the myelin sheath. So if you cut through the myelin sheaths, the internal structure of these myelins is very interesting. It's encapsulating the core, which is the axon, and around this core it has this concentric tubular lamellar structure with the spacing which is around tens of nanometer, and the entire diameter is a few micrometer and the length is a few millimeters. So in terms of both theoretical and practical point of view, it's a very it's a very interesting structure, and there's been lots of research on structures which resemble these kind of neurological myelin sheaths, and typically in physics and biophysics we refer to them as myelin figures. If we look at the bilayer structure within a myelin sheath, what we see is that the bilayers are formed of similar to many other biological membranes, are formed of phospholipids and glycolipids. And if you're an engineer like myself, what you would be tempted probably to do is to simplify the situation and maybe even oversimplify it and say that, okay, this is just an amphiphilic molecule which has a hydrophilic head group, which is the red ball in here, and has a hydrophobic tail. The tail is interesting though, because it has a multiple tail here, 2 tails, hydrophobic tails. And we know that both in biology, in other biological and synthetic materials, we have similar surfactants as the surfactant molecules. And for example, there's a beautiful experiment done by Sakurai and co-workers in 1989, where they picked up another phospholipid from egg yolk, and which has the choline group incorporated into its head group, and they basically packed the concentrated solution of this phospholipid into a small cavity, and they performed a contact dilution experiment where you just put the very concentrated self-assembly of these molecules in contact with pure water. And within minutes of the experiment, what they observed was that there was this growth of this tubular structure out towards growing towards the pure water solution. And if you look at these structures under a cross-polarized microscope, they look like live, worm-like structures that move a lot. They go through all sorts of instability, buckling, and twisting. And they have, you can clearly see that the center of these I'm going to refer to them as myelin figures, is different from the surrounding. And this is because if you cut through these, if you put these under electron microscope and cut through them, you see that the core is formed of, here especially, is formed of water, and the surrounding is basically very similar to the myelins that we just saw on the last slide, is formed of tubular lamellar bilayers. And so in my talk, I'm going to introduce a similar yet different system that allows us to grow these myelin figures in isolated form, and going to look at the dynamic growth of the myelin figures and try to persuade you that most of the growth mechanism, at least, even though these are non-equilibrium structures, they is mostly governed by the thermodynamics equilibrium. So what we know so far about the synthetic mimics of the myelins, which are called myelin figures, is that they are made up of these amphiphilic molecules that typically have two or branched tails. And the amphiphilic source is typically in a lamellar phase, where it's a planar, or most of the experiments starts with a planar lamellar phase and is soluble in water. And the growth of the myelin figures is often initiated at the highest concentration of the hydration or surfactant concentration. I'm going to pick a system that I believe is an interesting system for creating these kind of myelin figures experimentally. It is the molecule is of the very famous surfactant linear alkylbenzene sulfonate, which is a very commercial surfactant. In fact, probably all of you have come across it when washing your running your washing machine, because it's in all laundry detergents, and it has the hydrophilic head group, a benzene group, and then a branched tail, hydrophobic tail. And as other surfactants, it is lyotropic, meaning that if you increase the concentration of the surfactant, you will change the phases of the self-assembly of this molecule. You go through spherical, then elongated ellipsoidal micelles, and then a liquid crystalline phase, which is here a lamellar phase. The other interesting thing about this surfactant, at least in the range of concentration and temperatures that we're looking at, is that it's thermotropic, meaning that if we look at the temperature concentration phase map of this surfactant, the boundary between the phase, between the isotropic lamellar phase, which is in here, and the isotropic and the mixed lamellar and the liquid crystal, and the mixed isotropic and the liquid crystalline phase, which is basically the boundary between these two phases, is inclined, meaning that you can cross it by either changing the concentration at a fixed temperature or by keeping the concentration constant and just changing the temperature. And if we do not need to change the concentration, just change the temperature and go through these different phases, it allows us to keep the system in sort of at least quasi-static situation. And at the same time, also changing the temperature, controlling all the dynamics of these phase transitions via temperature, it allows us to perform some additional experiments that I will show later. The other interesting thing about this surfactant is that this phase transition from the micellar to the liquid crystalline phase is happening at rather smaller concentration of surfactant. And this lamellar phase, it spontaneously forms into a multilamellar droplet that will be the source of the myelin figures that I will talk about in this seminar. So as I mentioned in the first slide, these structures are multi-scale. So I will, in my talk, switch continuously between optical microscopy analysis, which gives access to dimensions, microscale dimensions, and small angle scattering, which gives us access to dimensions above tens of angstrom to thousands of angstrom. The first thing to remark is that this transition from the micellar to the liquid crystalline phase is isotropic. So if we start under the polarized microscope from the isotropic phase, the images are completely black. When we cool down the sample, we start getting some rare nucleation spots, and then you start seeing all these bright stars, which are basically our droplets. And upon heating, they disappear, and we can repeat this cycle and always get the same kind of structures. And these droplets are a few microns. There's no scale here, but this is around 5 microns, and they have a they're birefringent and they have a very clear maltese cross structure, which is the characteristic of multilamellar, spherical multilamellar vesicles. And so in parallel to these experiments, we can instead this time do the small angular scattering using neutron. And what we see is that if we start in the isotropic phase, what we have is a curve intensity versus Q is a typical curve for a micellar solution, especially charged micellar solution, which has a broad peak in the mid-Q region. So on this graph, just to note that when we move for larger values of Q, we are looking at smaller features at smaller dimensions. And when we move to the left, we are looking at structures at larger dimensions. And if we cross the boundary, you see clearly that there is a peak appearing, very short peak appearing at values of Q around 0.2 that is associated with bilayer spacing within these droplets, and then an upturn in the lower Q region, which is corresponding to this larger structure of these objects. And as we go deeper into this, below this boundary, then we start getting this this peak is rising, and also the upturn. And we can associate this, the location of this peak, to the bilayer spacing, as I just mentioned, and it's around 3 nanometer, the lamellar spacing, and we can confirm this with real space imaging under cryo-TEM, and you can see that the spacing between these lamps. So this is the image of one single droplet that's bounded by other droplets. And so, as I mentioned, when we do these experiments with temperature control rather than concentration control, there is another handle experimentally that helps us to control the experiments, and that is rate of cooling, for example. If we start the experiments at a specific temperature and end the experiment at a specific temperature, which is TF, but take different pathways to get there, different cooling rates, we see that the microstructure, this is under an optical microscope, the microstructure of the droplets that are formed are completely different. At higher rates of cooling, these are the experiments that I will refer to as isothermal cooling, there's a large number of nucleation sites, and the droplets are quite monodispersed, while at lower rates of cooling, we have polydispersed droplets that generate these kind of myelin structures. And I will mostly focus in terms of the optical microscopy on these structures, which are elongated, elastic myelin figures. So I said that in the at microscale, we have all these diverse kind of droplets that we get depending on the rate of cooling. And we did repeat the same kind of experiments on the in scattering mode, in small angle scattering mode. And what you see here is, so we are taking different cooling and heating samples. And what you see here is at different locations of this curve, is that, for example, at location 1, 3, 5, where we are supposed to be in the hot zone, where we are supposed to have the micellar phase, you see that the micellar phases, even after cycles of cooling and heating, they're always identical in both, in terms of both population and the size, and everything about the micelles is identical. The lamellar phase, which is achieved at equilibrium after cooling at different rates, is also identical. This means that if we pick a point in this part of the diagram where we are in the micellar plus lamellar structure, the concentration of the both phases in this at this location is going to be at equilibrium. So the amount of the ratio between the micellar and the lamellar phase at this location is just defined by the temperature and the concentration of the surfactant. And the other interesting point is that depending on the bilayer spacing that we get for these structures, even though at microscale they look very different, at nanoscale the bilayer structure that we get is very similar, and it's not dependent on the rate of cooling, which means that in the myelin figures, the structure that we have is very similar to the structures that we have in the droplets. And so if you look at the dynamics of these myelin formations in videos where in optical microscopy images, where we look at the sequence of images in which the droplets or myelins grow. We have actually measurements of also the droplet diameter growth, but I'm going to focus mostly on the myelin figures. So you see that at some point there will be, when the droplets connect to one another and not coalescing, but join one another, there are elongated objects like myelin objects that start growing. If we measure the length of the myelins, it's one of the most practical and simplest experimental measurements that we can perform to sort of understand the growth, the mechanism that that defines their growth. And we see that the length of the myelins is growing linearly with time, and its diameter is relatively constant, at least within the resolution of the optical microscopy. And so we can, if we estimate the volume of the material that's being added to this myelin during this growth, you see that because the length of the myelin is linearly growing with time and the diameter is constant, that there should be a constant volumetric rate that's being added into the myelin continuously. So in order to verify this hypothesis, what we can do is to go back to the SANS experiments and this time try to calculate the population of the micellar phase that we have at every point of time. So these are time-resolved SANS experiments where we cool down the sample, and at every time we calculate and we estimate the volumetric population of the micelles that we have. And if you look at the last graph here, you see that when we start the cooling, as we cross the boundary, we start with a constant concentration, end with another constant equilibrium concentration, and VI here is the concentration of the micelles, volumetric. But between these two states, we go linearly, which means that we are losing the volume of the micelles that we are losing is linear in time, which is the source that is feeding the myelins. And so if you repeat those experiments at different rates of cooling, the SANS experiments at different rates of cooling, once again, you see that it starts, we start at the same concentration of the micelles and with the same concentration of the micelles in equilibrium. But in between, we always keep the linear trend, the linear transformation in terms of the volume. But the rate is becoming faster when we cool down the solution at faster rates, at higher rates. And so here you see that if for a certain volumetric transformation from the micelles to the lamellar phase, we know that if we increase this rate, we are expecting to see an increased rate in the growth of the myelins. I'm going to call this the slope of the growth of the myelins alpha m here. And you can also clearly see that if we have larger myelins, in the sense that if they have a larger cross-section, they should be growing. If the volumetric transformation of material into these myelin figures is constant, we should have a slower growth. So we went back to the optical microscopy measurements, and what we did this time, we looked at the growth of myelins with different diameter of the base, and we looked at the slope of their growth rate. And what you see is that clearly when you have larger diameters of myelins, they grow much slower. And it nicely correlates also with the inverse of the square root of inverse of the diameter to the power 2. And the other thing to note on this graph is that if we increase the cooling rate by 10 times, the growth of the myelins is also increasing. The rate of the growth of the myelin is increasing by an order of magnitude, which is a proof of at least the hypothesis that we had for the constant volumetric rate of transformation from the micellar phase to the lamellar phase that controls this growth. So I'm sort of done with my talk in the sense of the equilibrium and the statics. I just want to show some of the nicer videos which are under dynamic conditions. The first one is droplets that are formed under static condition by cooling. And next to it, we have a condition where we are cooling the solution, but shearing the solution at the same time. And you can see that in the direction of the shear, instead of getting spherical droplets, we get elongated myelin figures. And these are once again, not equilibrium structures. So they start from a spherical droplet structure, which is a parent structure, and they grow in length. And at this point, if I stop the shear, they will retract and get back to a larger droplets, which is basically getting back to the original structure that they had. These three videos, they showed some of the dynamics that the cooler dynamics that these myelin figures have. The first one is a buckling stability that then it retracts and gets back to the droplet. Second one is a myelin that's growing, but at the same time has this helical motion, and it's also growing in length. The last one, which is probably my favorite, is, it's going to go on repeat, is a tiny myelin figure that is originated at the, wait for it to appear again, is this tiny myelin. At the beginning, the contrast is not too good, that's swimming upward, changes mind, takes a U-turn, and swims backward. And meanwhile, in the solution, you see tiny myelins in other locations that do similar kinds of swimming behavior. So they're very, although I just looked at in this talk, I just introduced their statics, they're very dynamic, and especially under flow, they do have very interesting behaviors. With that, I would like to finish my talk, and I'm happy to answer any question if I can.